Thank you, Peter, for the introduction. And also, I want to thank ILSI North America for inviting me to be here this morning. And as Peter mentioned, my job this morning is to kind of give you guys a 30,000-foot view of genome sequencing, and then we're going to follow up with some examples for those people who are working with it very much in a real-life setting. So DNA sequencing began in the late 1970s, and the first molecules that were sequenced include viruses and phages. The Sanger sequencing method, which is, continues to be the gold standard of DNA sequencing, was developed in the late 1970s and was the workhorse of DNA sequencing for over 20 years before next-generation sequencing methodologies were introduced. The Sanger method, in terms of using that for genome sequencing, involves what we call shotgun cloning. Basically, we would take pieces of DNA, we would take the genome of an organism of interest, fragment that, and then we'd clone those pieces into E. coli and amplify the pieces within E. coli. So as you can imagine, this is very labor-intensive and very time-consuming and also quite expensive. The first bacterial genome, Haemophilus influenza, was sequenced in 1995. The genome size was about 2 million base pairs. And just to put things kind of into perspective, my first semester of graduate school actually is when the first Listeria monocytogenes genome sequence was published by the Pasteur Institute from France, and that was basically sequenced using the Sanger method. So that opened up a world of opportunities for us. I can remember how excited we were back in 2001 that we could design primers to, develop multi-locus sequence typing schemes, and that was a big part of my PhD work. And then in 2003, the first human genome sequence the finished human genome sequence was released. And that project, many of you are probably familiar with that. It took about 13 years and cost about $3 billion. Even today, using the most advanced Sanger sequencing equipment and protocols, it would still take about 3 years to sequence a human genome. So obviously, when we had this information, we realized that we needed more genome sequencing, and there was a big push in order to develop some next-generation sequencing technologies. And some of the incentives that were put behind this included Craig Venter Science Foundation had promised $500,000 to the first group that could produce a technology that was able to sequence a human genome for $1,000. So all of these things are sort of, behind the race for a $1,000 human genome. The X Prize Foundation promised somewhere between $5 to $20 million for the first technology that was capable of sequencing the human genome for $1,000. Also, the NIH put a lot of money towards research grants that could be used towards sequencing genomes for, more feasible cost in terms of being able to do some population studies. And then in 2005 and 2006, right about that time, the first wave of next-generation sequencing technologies were released. So now, some of the speakers later on this morning will talk more to this, but now it's possible to sequence a bacterial genome for less than $100. Even in my lab for research purposes, we're quite interested in sequencing Listeria monocytogenes genomes. One of my graduate students was doing library preps to put 30 on the machine, which would cost us, less than a couple of $1,000s. So we can really, do a lot in terms of being able to sequence genomes at this point. Commonly used platforms, Illumina, either the HiSeq, MiSeq, and then NextEra, which would be the next generation of sequencing technologies coming out. Life Technologies has the Solid, and then also the Ion Torrent. And then the first instrument to come out was the Roche 454. Also, we have PacBio and Helico Sciences as well. And so being able to sequence a genome for less than $100, that's putting us, sort of on par with the cost of doing PFGE subtyping of isolates. So I think, Peter's going to talk more this morning that we're going to start seeing genome sequencing replacing PFGE as the gold standard of subtyping bacterial isolates. So next generation sequencing technologies, sort of just grouping them all together, what they do, able to generate a lot of data very quickly. So as I mentioned earlier, we can actually, in my lab for research purposes, we can take bacterial isolates, take them downstairs, do our library preps, get them run on the machine, assemble the data and annotate that. We can basically have information within a week, which is, sort of on par with, taking 2 or 3 days to do PFGE typing and then the analysis after that. So there's different chemistries and platforms. How they differ from each other is basically, how you prep the DNA library in terms of making the template, and then also how they're actually sequenced or imaged. So in terms of template preparation, you can either, a lot of the technologies basically clonally amplify pieces of DNA in clusters, or some of the newer technologies are actually able to use a single copy of DNA as template. In terms of the actual sequencing and imaging, you either have reversible termination, that's the Illumina technology, adding single nucleotides at a time as a substrate, sequencing by ligation, that would be the solid technology, and then also real-time sequencing, so that would be PacBios or Helicose. So in terms of the cost of DNA sequencing, you can see here that when we introduced next generation sequencing technologies, you can see from the late 1980s to the early 2000s that the cost of Sanger sequencing was substantially reduced. But when we introduced next generation sequencing technologies around 2005 or 2006, you see a very steep decline in the actual cost of sequencing. So as I mentioned before, some of the commonly used technologies, the first one to come out was the Roche or 454 Pyro sequencer. Illumina has either the MiSeq or HiSeq, and also Nextera. Applied Biosystems has the solid, and then also the ion torrent. And then we have Helicose and Pack Biosciences as well. So we're just going to talk briefly about a couple of these different technologies. If you guys are interested in learning more, talking more specifically, we can talk after the session today. So this was the first technology to come out here, the 454 Pyro sequencing. And basically what happens is you take fragmented DNA and then you ligate that DNA to adapters, and then within basically a well in a microtiter plate, the DNA is actually amplified clonally on a bead here by emulsion PCR, and then you pass the different nucleotides, the substrates, over the well, and if whichever one is next in terms of sequencing by synthesis will bind. The limitation with this technology is that you read the fluorescent signal, and then sometimes it's hard to tell. Let's say if you have 3 Ts or 4 Ts, it's hard to tell the difference between that. So there are some problems in terms of fidelity of particularly homopolymeric tracks of nucleotides. And the benefit of this particular technology is that it has, the longest sequence read. The next one, Illumina. So basically either the HiSeq or the MiSeq, the technology is very similar. We have basically bridge amplification here. So similar to the last technology, you start out with DNA molecules and then you ligate those to adapters. And those adapters are basically going to have your DNA become mobilized to a fixed surface, which is a flow cell. And within that flow cell, you start to get clonal bridge amplification here. You can see that you get these clusters of the same DNA molecule that are basically amplified in the flow cell, and then you're able to read the nucleotides that are added to that by a fluorescent signal. Just one more technology to talk about briefly here, the solid, which is manufactured by Applied Biosystems. Again, starting out with the same sort of concept here. You take fragmented pieces of DNA and you ligate them to adapters. You can either have fragment libraries or mate paired libraries if you wanted to have longer reads, which makes assembly easier. You add that with polymerase, and then your DNA molecules are amplified on a bead. And the interesting thing, or I guess one of the main benefits of the Applied Biosystems solid system is that your probes, your primers that you're working with, they have 2 nucleotides on them and they shift over a base at a time. So the integrity of your sequence is very high because basically every nucleotide that you're sequencing gets probed twice by the same primer. So you have less problems in terms of SNPs that you find. You have more confidence that they're true SNPs and not just an artifact of DNA sequencing error. So this is just a table to kind of briefly show comparison of some of the new next generation sequencing methods as they compare to the gold standard Sanger method. So the ABI 3730, that's the Sanger sequencing here. And when it was launched, the reads here, and then you can compare that to the different pieces of equipment. So you can see the amount of data that you get from next generation sequencing technologies is just so much more, it's substantially more than what we could ever do with Sanger sequencing. And it's not to say that Sanger sequencing is not useful at this point anymore. I mean, we still use it for research purposes in terms of being able to sequence genes, and then also for SNP typing and those type of things. But it's, in terms of sequencing an entire genome, it just, makes a lot of sense to use these newer technologies. And at Texas Tech, in our core facility, we have actually the MySeq, and I think that's something that's pretty common. The instrument you can purchase with the instrument and the software and everything altogether, the whole package, for less than $100,000. So it's something that you could actually have, in an individual research lab. So I'm just going to go through a couple of different examples of how, what we can use next generation sequencing for. There's a lot of different applications. I mean, the most obvious would be just de novo sequencing of isolates that you're interested in. And we do that on a routine basis, but you can also use it to study the transcriptional profile or transcriptome of organisms that are being grown in certain conditions. Just recently in our laboratory, we were using RNA sequencing to understand what genes are turned on in salmonella when it's attached to romaine lettuce as compared to stainless steel slides. And we got some really interesting information and preliminary data that was used for a grant proposal. So that's an application. Another application would be metagenomics, so we can take a food sample or a rumen fluid sample or whatnot and be able to understand, what's the population of isolates in a very complex environment based on deep sequencing of 16S. Also can be used for epidemiology, and I think Peter's probably going to give some really good examples of that, but I'm going to briefly this morning speak about some genome sequencing that was done through a collaboration to understand the short-term evolution of Listeria monocytogenes in a food processing plant, and then just give an example, the first example that I'm aware of where whole genome sequencing was actually used as part of an outbreak investigation. So in the first case study, this is a Listeria monocytogenes strain that was isolated from a food processing plant that was implicated with a sporadic case of disease in the late 1980s, and then was also associated with an outbreak in 2000. So in this particular study, what we did is we took an isolate from human disease, or a human clinical isolate, and then also a food isolate from the 1988 incident, and then also a human and food isolate from the 2000 outbreak, and we sequenced those to understand if it was the same strain, because the strain from the case and the outbreak had the same PFGE type and the same ribotype. That doesn't necessarily mean that they're the same clone or they derive from the same ancestor. So we wanted to understand how that particular strain had evolved in that processing plant over the 12-year period. And this is just the title of the paper. So in short, when and this sequencing was done using the first technology of next generation sequencing, the 454 pyro sequencing. So this is a little bit of data on the actual genome sequences that we got. And a contig is basically just a continuous sequence. And then when we think about a genome, let's say Listeria monocytogenes is 3 million base pairs. When you sequence that, it doesn't come out all in one piece. Sometimes you may actually have thousands of contigs, and it's not really that important to get them in the exact order, but you put them together in what's known as a pseudochromosome or a pseudomolecule in order to analyze the data. So this is just some statistics here, and now when we do next generation sequencing, we're actually in the era of getting hundreds of fold of coverage on these genome sequences that we can assemble. So when we look, the genome sequences were, very typical of Listeria monocytogenes, around 3 million base pairs. And interesting, we had sort of one backbone, one major chromosome molecule, and then also 2 prophage, which a prophage is basically a bacteriophage that becomes inserted into a bacterial chromosome. And one of the prophage was inserted into COMK, which is thought to have some sort of regulatory purposes in Bacillus, but it's not actually active in Listeria monocytogenes. And this particular prophage, through more recent studies, has been associated with Listeria monocytogenes that actually persist in the environment of processing plants. And then also another prophage into the tRNA gene. So looking at the genomes, there were about 40 SNPs, or single nucleotide polymorphisms, observed in a single isolate, and most of these SNPs were actually found to be an artifact of actual errors of the sequencing. When we look closely comparing the genome sequences of these isolates that were associated with this plant, that are highly related. So there were only 8 SNPs. 2 of them were unique to the human clinical isolate, and only one of the SNPs was actually found in an intergenic region. So basically we saw that, looking at these isolates persisting in this plant over this period, only a few single nucleotide polymorphisms and some recombination events here shown through this figure here had occurred in the COMK gene. So the isolate had been very conserved over this 12-year period. So looking at this actual sequence information, there were a lot of indels. Indels are short for either insertion or deletion mutations. And as I mentioned earlier, most of these actually were identified to be an artifact of the sequencing technology, and they weren't actual mutations. So over the 12 years, basically we started with an ancestral genome sequence here, which was split into something that was associated with human disease and something that was isolated from food. And we had accumulated, one SNP in the human isolate and 4 SNPs in the food isolate. We had these recombination events in COMK and a tRNA prophage mutation or recombination. Then we had this secondary ancestor. And then after that, when we looked at the isolates from the 2000 outbreak, we had 8 mutations that occurred in the food isolate and then 1 mutation that had occurred in the human clinical isolate. So in summary, it's, looking at this in a natural environment, these mutations that have occurred, it's very different from the evolution that we could experience in a laboratory setting. In a laboratory setting, we're, sort of pushing bacteria to multiply as quickly as they can, and we can actually see, mutations that would arise even through an overnight culture. It's quite easy to make an organism resistant to antibiotics just by growing them in the presence of antibiotics. And we saw that the same Listeria monocytogene strain persisted in the food processing plant environment for 12 years, accumulating very minimal changes. And this was sort of one of the first examples of using next generation sequencing to study the evolution of foodborne pathogens in a processing plant environment. And we saw that the next generation sequencing technologies are obviously quite fast and efficient and generate a lot of data, but you also need to interpret that carefully in terms of particularly looking at single nucleotide polymorphisms. So the next example that I'm going to share with you this morning is the first example that I could find where next generation sequencing was used as part of an outbreak investigation. And I think our speakers later on this morning will have several other examples. I didn't want to take any of their stories, so we'll stick with the basics here. So this was an outbreak of listeriosis that was associated with deli meats that were produced by a facility in Canada. The outbreak contained 22 deaths and around 60 illnesses. A majority of the isolates associated with this outbreak had the same pulse field gel electrophoresis pattern. But when we talk about PFGE patterns, some patterns are more common than others. And just if you have an outbreak that's associated with a very common pattern, it can be difficult to find the food vehicle that's responsible for disease, just because of the nature of that pattern being common. Looking at the isolates associated with this outbreak, one isolate had a different pattern, and it differed by 2 bands. So when we think about PFGE typing, we say if it's less than 3 bands difference, then we say they're possibly related, but that doesn't necessarily mean they're related. So in this case, genome sequencing was used to determine if these isolates that had the 2-band difference were actually part of the outbreak. And this particular example also was using the 454, the first sequencing, next generation sequencing technology that was introduced. And then all DNA sequence were closed actually in this case by the Sanger method. So you can see that we had 40 to 70x coverage. That just means every fragment in that genome sequence was sequenced either 40 to 70 times. You have to have that depth of coverage to be able to assemble the information. So the chromosomes were very similar to each other. They were collinear. But looking at the isolates associated with the outbreak that had slightly different PFGE patterns, only 28 single nucleotide polymorphisms were observed, and 3 insertion-deletion type mutations. And then also, interestingly, again, another prophage, a large prophage that accounted for the differences that were actually observed in the PFGE patterns. So with that, I just want to do a couple of acknowledgments here. I would like to thank Martin Wiedemann, Renato Orsi, and Hank Bakker from Cornell University. Some of these slides that I presented this morning were from their research, and also collaborators from Life Technology in terms of these projects that were presented, the Broad Institute, and the USDA for funding.